pleasure for me personally to be here again and to be at, I would say, first in-person meeting since long time ago. And, so we've been doing this for quite a bit of time, and I have a lot of colleagues, obviously, in this hall who became good friends over the years. So as I already said, it's a big pleasure to be here and start new round of CASP and have this in person. And while I'm still waiting for my slides, I want to say that John just outlined the perspectives and maybe where we are going with protein structure prediction in future. I'll be more talking about nuts and bolts and about organization of CASP, what happened with participation, targets, and then second part of my presentation will be on the domain definition and domain categorization, which is very important because everything that you see on the web pages, and I hope that you already had a chance to look at those and see the results, is based on the domain definitions and domain categorizations that we've done this CASP together with Dan Rigdon, who is our assessor for tertiary structure prediction. Obviously in previous, here are slides. In previous CASP, we had a benefit of having Lisa Kinch from Nick Grisham Group. And as you know, she is very knowledgeable in structures. She works on ECOD domain classification, so she was very helpful in defining domains and categorizing them. And this CASP, she was not with us, and we had to come up with new ideas with Dan how to define domains and categorize them, and I'll be talking about this in the second part of my presentation. So all the numbers, all the data about this CASP experiment, about its conduct, are on the web. So if you go to CASP15 numbers CGI, you'll see the data. And I still cannot advance this one. So now talking about the groups. After terrific performance of AlphaFold 2 in the previous CASP, we were afraid that we'll see a big drop in the participation for this CASP. And in fact, there was a drop, but not a big one. It was a really slight drop. But at the same time, there was a big increase in separate categories that people found it interesting. And that's when you look to the graph to the right of you, the yellow graph. So you see that's participation in quaternary structure prediction, in assembly prediction. So it was more than twice increase in number of groups participating in this category, and that was really encouraging for us. And when you look at the number of groups overall, so we see that was a small drop, but don't forget, as John said, that we got rid of 3 different categories. Got rid of contacts, we got rid of quality assessment in the form that we used to have before we started working with multimers, then assisted prediction. So all these obviously lowered the number of groups, but still participation is very healthy. And also we started with 2 new pilot experiments, which are RNA and ligand prediction. And we just wanted to see what's the interest among the predictors to these categories, and interest was extremely high, as I should say. Probably assessors would tell their opinion, but having 42 groups for the first experiment in RNA and 33 groups in ligand category, that was very impressive. So we had 89 prediction centers from 17 countries participating. So that's number of groups and number of prediction centers are slightly different things, right? So, and this time the parity was between the United States and China. We had approximately the same number of groups participating from 2 countries. So number of predictions now, we have approximately the same number of predictions in 2 CASPs. But again, if you look to the right on this yellow graph, so you'll see that number of predictions in quaternary structure increased a lot, and this quaternary structure prediction will be a highlight of this CASP as you'll hear later in tomorrow's presentations from ESGI. So it's really interesting results there. Now, once we collected all these predictions, we started evaluation, and we have over 30 different evaluation tools at the Protein Structure Prediction Center. And we ran all of them, generated millions of scores, and then we organized these scores and then presented them to assessors so that they can make sense of this huge amount of data. So we had 400 gigabytes of data generated in CASP in terms of models, results, and so on and so forth. We were really helpful. Some of the tools that we never had in CASP were really helpful. For example, without SWORD, we couldn't do domain definitions in a more educated way with DAN, because, let's say, D-domain or domain parser cannot handle big complexes, and we had proteins that had over 1,000 residues, 2,000, 3,000 plus residues. So only SWORD could help us doing that. Then FoldSeq, it's an amazing program that runs very fast and finds structural similarity among the PDB. So in previous CASPs, looking for structural templates was a big bottleneck for us, because they had to run it in 4 or 5 different stages. And now you just run FoldSeq, and then in a matter of minutes, you have candidates for the best templates, and then you just run it with LGA, and you get LGA scores, what we used to have in CASP. So it was really instrumental and nice. And then some other programs, RealLG from Randy Reed, US-Align, which is improved MM-Align from Yan Zhang's group, where they fixed some problems with fitting of chains between model and the target. And then ACE score that was written by me some time ago, but it was written based on the distances, and now we work with the predicted LDDT values, so it was refurbished for this purpose. And then QS score, and that's from Gabriel Studer in Torsten group in Basel, and they also improved fitting between the chains in different structures, and implemented DocQ score into the package. So all these were very useful for us in CASP, and all these data are reported already on the web. Okay, so we had 48 structure determination groups giving us, but I believe that's not the next slide. Okay, so this is the next slide. So we had 96 targets, and we always strive to have as many structures as possible. John always wanted 100 plus. From my experience, when you start getting structures and analyzing them and looking at models, because it's always interesting. So I noticed that after having first 20, 25 structures and evaluating models on these structures, you already have a pretty good idea who will be good in this CASP and who will be not that good in this CASP. So in my opinion, so having around 40, 50 structures is enough to have good and rigorous testing. But obviously, the more the better. And this CASP, we got quite a bit of structures, 94 structures, which is a really good amount. And also, when you look at the number of structures in quaternary structure prediction, so that more than doubled. So we jumped from 20 to 47. So that was also contributing to the rigor of the assessment that ASGI and Purju did in assembly prediction. Now, let's look at the RNA and ligand. So that's also impressive. When you just look about 12 and compare it to 94, it's maybe not that impressive. But people who are in RNA business, so they know that these targets don't come easy, right? So having 12 targets in RNA, so it's kind of an achievement. And we had these 12 targets and gave it to people for prediction. So that was also really nice. So that's our geography. So if you just think 48 structure determination group from 14 countries. So actually we have a very wide network of target providers, and we still work with these people. So we were lucky somewhere in the middle of this process, like CASPs 3, 4 CASPs ago, when we had these structural genomic centers generating hundreds and hundreds of structures, and we obviously benefited of those structures. And then when it dissipated, so we had to go one by one and cherish all the predictors, all the experimentalists. And because of that, we are still able to get quite good representation of targets of different quality. And I guess that, in my opinion, and I'll be touching this a little bit later, targets in this CASP are coming quite difficult, so they appear to be really challenging to predictors, and you'll see this from the results that will be reported tomorrow. Combination of targets, not combination of targets, but determination methods, how these targets were obtained, is quite different in CASP15 than it was in CASP14. So we used to have predominantly X-ray structures, and that's still the case in this CASP. So the vast majority of targets are X-ray structures. But when you look at number of targets that were solved with cryo-EM, that grew up substantially. So in this CASP, we had more than a quarter of all targets solved with cryo-EM, while we had around 10% in CASP14. And these targets are quite different, as you know. They are larger, they are of big complexes, quite often intertwined complexes, and that presents target difficulty. And we still have target difficulty in CASP, as I will be presented later, based on homology. But that's nowadays probably not the most important factor that determines how difficult is this target. Maybe length, maybe multiple sequence alignment, how shallow it is or how deep it is, so other factors. We have to figure out what are those, and maybe we'll hear from the assessors later on about that. So now look at the length of the targets. In CASP15, we had 21 targets larger than 500 residues and 12 targets that were larger than 1,000 residues, while in CASP14, we had only 3 of those. So obviously, big change in the target composition. And now I'll be talking about domain definition and categorization. As I said, we did it together with Daniel Rigdon. So once the target were released, or we got the target from the experimental list, we prepared it, and then we ran search for templates with sequence-based methods, with structure-based methods, and then we tried to do some original domain definition using the programs that we had at hand, and then we compared one with the other and came up and were coming up with the original definition of domains, run an evaluation, check in some data, Gaussian plots, and I'll be talking about Gaussian plots next. And then, if needed, this was reiterated again until we converged and we had final domain definitions. So there were some cases where we did not have to choose. So the data choose for us. So what should we do? For example, this is the target 1120. So now we are starting naming targets and going back to CASP. Target 1120, that's DNA binding protein. And it's a monomer, but it had 2 chains in the symmetric unit, and it had different orientation of domains. If you look here, you see the cyan and the green one, and somewhere in the middle of this helix at the bottom, you see that it deviates quite substantially. So the data told us that we have to cut it here. And that was domains 1 and 2 for this target 1120. Another example, 1121, that's a DNA cleavage protein, and it's a homodimer. So that's how the target looks like as a dimer, right? But when we superimpose chains, green on the cyan, we get something like that. So if superposition is anchored to the right-hand side of this target, so you see that left-hand side deviates quite a bit between green and cyan. So when you cut it here, so again, you get quite tight superposition of chains 1 and 2 in the target, and those were used as our reference structures, domain reference structures for this target. Another case where we can call it have to split. So that's target 1170, that's the holiday junction. That's a hexamer with non-crystallographic symmetry, and some chains deformed to accommodate DNA, as you can see going inside this hole in this complex. So there are 6 chains. So if you superimpose them on the top of each other, you will see something like that. But if you look more attentively on this, so there are 4 chains here, and then 2 chains here. So 4 2. So you see they're quite different in the top portion of this target. So essentially when you cut it here, so you'll get the 2 domains for this target 1170. So that's what we've done. And now something that you can see at the top of the slide, which is called to split or not to split. So those were subjective decisions, but obviously subjective decisions that were promoted by analysis of the data and analysis of the primary evaluation. For example, in this case, for target 1124, when we run the domain, it says 2 domains. So, and it's clearly 2-domain target, as you can see. But when you look at the Grisham plot, you can see that all the points line along the diagonal, and that's why it's 1 evaluation unit in the end. For those of you who don't know what is Grisham plots, so this kind of plots was introduced by Nick Grisham in CASP9, I believe, and it tells you how different are prediction of the whole protein or combination of domains compared to separate domains. So on the x-axis, you have GDTTS of the combined unit, so 2 domains, let's say. And on the y-axis, you have GDTTS of separate domains, weighted, and then averaged. So essentially, if people are getting equally well domains and the whole thing, you'll see points along the diagonal, right? If points are above the diagonal, it means that people are getting separately domains better than they are getting the complex. So in this case, it's probably beneficial for evaluation purposes to split. But in this case, there is no need to split, as you can see. A little bit more complicated case is target 1112. So here again, you can see 2 domains. You run domain parsers, it tell you, it's 2 domains. You look at the Grisham plots, and here is a little bit more complicated case. So you can see a band of points going off the diagonal. And then you see quite a big bunch of points that are sitting right on the diagonal. And these are good predictions. These are predictions with high scores. So essentially what we've done in cases like that, where we had both, so we went with the majority. So if a majority of the most dense population of points, of models, were on the diagonal, then we didn't split. And if the vast majority of points were off the diagonal, we did split. So in this case, vast majority sits right on the diagonal. So decision was not to split 1 evaluation unit. Now we go to more complicated cases, more than 2-domain proteins, and that's target 1154. That's a Slayer protein. If you look at it, visually you just would say it's 4 domains. You'll run D domains, 4 domains. But then you look at the Grisham plots, and the top 2 plots, that's about domain 1, which is, I'll try to do this. Oh, now it works. Perfect. This is domain 1. This is domain 2, 3, and 4. So you see that domain 1 does not like domain 2 and domain 4 to go together. So these are these 2 plots. But at the same time, domains 2 and 3 go well together, and domains 3 and 4 go well together as well. So that's why what we've done, we started with 4 domains and ended up with 2 evaluation units like colored here. So that was evaluation unit 1, and that was evaluation unit 2. And amazingly, I have to emphasize here that both of these evaluation units are free modeling targets, which probably 2 CASPs ago would get scores of 30 or 40 GDTTS. And now when you look at that, it's like GDTTS of 97, that's practically the experimental structure, and here you have GDTTS of 87 as perfect predictions. And when you look at the Grisham plots for this kind of targets, so you can see that it's definitely 2 different big angle between the diagonal and the trend line of the points, and that's why that was the final definition of domains for target 1154. Another interesting case, and they have 2 more for you. So this one is target 1158, and that's an ABC transporter, and it opens and closes, so it breathes. And it also has different ligands inside. So, and here, if you run any domain parsers, you get very conflicting data. So one tells you it's 2 domains, but it's consecutive domains, right? So if you look at this, So you have 48 to 1022, and then just small domain at the top. But when you just follow how this target goes from N to C, so N is blue and C is red terminal, so it goes like this: blue, 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 then it switches here, light blue, and go big again on this side, and again on this side, down here, and ends up here. So if you try to define this sequential as, for example, D domain made, so it makes not much sense. And also SWORD has 5 domains for that. So we came up with then with something that makes functional sense, and that's domains defined as the protein function. So actually it opens and closes like domain 1 and domain 2. And if you look at the domain definitions, obviously it's not sequential, far from being sequential. But people predicted it quite well. If you look at the Grisham plot, so you can see that the separate domains were predicted to GDTTS of 85 or something close to 90, while the whole thing was predicted much poorer, around 60 or something. So that was our final definition, and it was a 2-domain target. And the last case that I want to show you is a target 1169, so that's 3,000 plus residues target. So when I spoke with the person who solved this structure, so he said that that's probably the longest single chain target protein that was ever solved. So I believe him, what he told me, but that's really huge target and interesting for prediction. And here is just all things of complication, It's better propeller here, so when it goes here, then goes here and returns back here, and then it goes here and ends up somewhere here. So it's really hard to even imagine, this topology and how this protein is assembled. We got an advice from an experimentalist, how he sees the domains, and then we started with 7 evaluation units on this target. And again, thanks to SWORD and developers of SWORD, because no other program could handle it, SWORD could. So, and takes quite a bit of time to do that, but still can do that. And then we started with 7 evaluation units, and then started joining, again, according to Grisham plots, and ended up with 4 evaluation units, like specified there, and colored in this part, in the lower part of the graph. Okay, so that's about domain definition, and the final portion is domain classification. And in previous CASPs, what we've done, we had extra parameter. We had homology, and we had performance. And now, in this CASP, performance is not a discriminatory parameter anymore, because everyone is performing well. So as I showed you in one of the targets, there were 2 domain targets, huge domains, free modeling, 90 plus predictions, GDTTS. So we decided to get rid of the performance and just look at the targets in terms of their sequence homology, not sequence homology, but sequence similarity to available structures, also structural similarity. So here LGAS on the x-axis is structural similarity, and here is sequence similarity in terms of AG search probability times coverage, and that's data from CASP15. So that's the categories, the different colors as they were defined in CASP14 with manual intervention and big manual thoughts, from Lisa Kinch. So here at the top corner, we have the easiest targets, these blue ones. Here we have the most difficult free modeling targets, that's black circles. And then we have some intermediate targets, this hollow black are FM/TBM, intermediate category. And this, whatever color it is, comes brown for me or orange. So it's TBM hard. So, and if you look at this graph, so you can kind of think about how would you split this graph based just on the homology to have different categories in different pits. So we tried many different ways with Dan, and we ended up with this. So it's simple and it's linear, right? So, and if you do this, you'll see that all the template-based modeling easy points are in this blue triangle. All the difficult targets are in the lower left triangle, and then the targets in gray zone are in 2 areas here, while all the, not all, the majority of the TBM hard targets are in this band, and the majority of the FM/TBM targets are there, right? So then we decided, okay, so let's take CASP15 data and do exactly the same classification according to these parameters. And that's what we've done. So that's the CASP15 data. So we just plotted all the points that we have in CASP15 on the same axis and did the same coloring scheme. And here are our CASP15 free model, template-based model in easy targets, free model in targets, and that's FM/TBM, not that many overlap compared to CASP14, and that's TBM/TBM hard targets. Again, that's the main point of doing this, was to check and compare with previous CASPs, right? Because as I pointed out, that there are other parameters that define now difficulty of targets. So there's the slide summarizing it, telling us that there are more free modeling targets in CASP15. And again, that may be an indication of harder targets in this CASP compared to the previous one. If you look just at the target difficulty in terms of sequence identity and coverage to the single best template that you can find in the PDB, so we can see that, so that's gradient going from the red to the green color there in the top corner. So that's the easiest targets, that's the hardest targets. So when you look at CASP15 free modeling targets and CASP15 free modeling /FM+TBM, which are these yellow squares, so we can see that both of these categories are probably the hardest among all the CASPs that we used to have. If you look at the sequence identity, so it's approximately the same in CASP15 and CASP14 for free modeling, and also this and this for FM+TBM, but coverage is lower in CASP15 than it used to be in CASP14. Okay, so we have a few more slides for you. On domains-based size information and size data is similar to what I showed you on the target, whole target base. So we have 19 targets in CASP15 that were larger than 400 residues, and only 7 in previous CASP, so larger targets, more difficult. And then the last thing that I want to discuss is ineffective, and I want to thank to Claudio Mirabello, who helped us with running AlphaFold 2 on CASP15 targets, and these predictions were released to all of you, so you know they were released through the server web page. And he had all the